In this video, I want to complete the discussion about the nephrons of the kidney. Once again, the nephrons are the functional unit of the kidney, which means they are the workhorse of the kidney, carrying out all the relevant work of the kidney in producing urine, secreting enzymes, and hormones. So let's get going on this. What we see in these two images right here, on the left, we see a sample of concentrated urine, and on the right, we see a sample of dilute urine. Concentrated urine is concentrated in solutes. Dilute urine is concentrated in water. There's more water than solutes on the right, more solutes than water on the left. Now, quick review, there's solutes, which are anything suspended in solution. So if we are making a glass of Kool-Aid, we pour water in the glass. That is the solvent. Water is a universal solvent in the body. Then we take the powdered Kool-Aid and we put it in. We take a scoop of Kool-Aid and we pour it into the water. The Kool-Aid, the powder, the little specks of Kool-Aid are the solutes. So anything suspended in solution is the solute. And the relationship between, or the ratio between the solute in the fluid or the solute in the water determines the concentration. So if we have put in 2 scoops of Kool-Aid, then there is a higher concentration of solutes. If we put in 3 scoops of Kool-Aid, there is yet an even higher concentration of the solute, or a higher concentration of the Kool-Aid. As the solute concentration increases, the water concentration decreases. So if we just had half a scoop of Kool-Aid, then we have a fairly low solute concentration and a much higher water concentration. And when we're looking at urine, the one on the right here, this urine sample on the right, it has much lower solutes than the urine on the left. And when we're talking about solutes within urine, we're talking about anything within the filtrate. We're talking about urea, uric acid, creatinine, ions, glucose, anything that was initially filtered and was not reabsorbed. Now, the question is, why are we trying to get rid of a bunch of solutes as we see in the urine on the left? It really has less to do with the solutes than it has to do with the water. And the concentration in the urine is really dictated by how much water has been reabsorbed by the collecting duct. So we're going to talk about the collecting duct. We're going to talk about reabsorption in the proximal convoluted tubule, and secretion and reabsorption throughout the nephron. Do we want concentrated urine or dilute urine? It really depends on the conditions of the body. If an individual is dehydrated, meaning they have low fluid volume within their extracellular fluid, then we're going to end up with more concentrated urine because the body, the nephrons, the kidneys are going to retain that water in the body by reabsorbing it from the kidneys back into the bloodstream. So we're going to look at all of this as we move forward, and let's do that. Okay, just review of the nephron that we see in pink here. We have the tuft of capillaries known as the glomerulus in light blue on the left, afferent arteriole leading into that, efferent arteriole leaving it. And it is lined by cells on each side. So this green line here is formed by simple cuboidal epithelial cells, and that is on both sides. And these cells are often referred to as tubule cells because they bind each side of that tube. As we move down the loop of Henle, the descending portion of the loop of Henle is often referred to as the thin segment, whereas the ascending portion of the loop of Henle is often referred to as the thick segment. And the difference is based on the type of epithelial cells forming the walls or making up the tubule cells. the thin segment of the loop of Henle is formed by simple squamous epithelial cells, whereas the ascending portion of the thick segment is formed by simple cuboidal epithelial cells. So the great majority of the nephron, at least from the proximal convoluted tubule all the way down to the collecting duct, is lined by simple cuboidal epithelial cells, with the exception of the thin segment of the loop of Henle, which is lined by simple squamous epithelial cells. So please keep in mind, all along the nephron, on each side of the lumen, we have cells, simple cuboidal epithelial cells, with the exception of the thin segment, most of which is part of the descending portion of the loop of Henle. And we'll discuss why we have simple squamous in the thin segment and simple cuboidal in the other regions. Okay, so let's take a look at the proximal convoluted tubule. This right here would be the lumen of the proximal convoluted tubule, coming from the glomerulus, which in this case we're going to say is on the left. So filtrate is moving from left to right towards the loop of Henle, which would be somewhere downstream on the right. And these are simple cuboidal cells. We can see a nice round nucleus pretty much right in the center of these cells. On the apical surface of these cells, we see surface modifications known as microvilli. Microvilli increase surface area. The more surface area a cell has, the more opportunity there is for things to move into those cells or out of the cells. So in this case, there's a lot of reabsorption happening within the proximal convoluted tubule, within the cells of the proximal convoluted tubule, or I should really say across the cells of the proximal convoluted tubule. Anything that's reabsorbed is going to pass from the lumen across these cells. And I know I always draw the peritubular capillary above the proximal convoluted tubule, but it's not a three-dimensional image. Things can cross these cells as well. So once again, this is the apical surface of these cells. This is the basal surface of the cells. We see the plasma membrane in green around each cell, and we see a ton of mitochondria within these cells. The simple cuboidal epithelial cells of the proximal convoluted tubule are packed with mitochondria because they utilize a lot of ATP to transport things in the process of reabsorption back into the bloodstream, back into the peritubular capillary, back into the extracellular fluid, which would be here or here. As a matter of fact, the proximal convoluted tubule, the cells of the proximal convoluted tubule are going to use about 6% of the day's use of ATP. 6% of the body's use of total ATP is happening within the proximal convoluted tubule. Why is ATP needed? And the simple answer to that is because things are being moved against their gradients. We've talked about diffusion, where things move from an area of high concentration to an area of low concentration. And certainly that plays a part in reabsorption. So as things move across from the lumen into the tubule cell, some things are moving down their gradients. But if it's moving down its gradients into the cell, it's almost certainly moving against its gradient across the basal surface. And this being in an anatomy class, I don't want to worry too much about the details on that. But at some point, the entities are going to be moving against their gradients. So let's just take sodium, for example. It's generally moving passively into the cell and then needs to be actively pumped out of the basal surface of the cell. When something is actively pumped, it requires ATP. So there's a ton of ATP used. Keep in mind that upwards of 75% of our filtrate, that is to say, 75% of the plasma that is filtered in the renal corpuscle gets reabsorbed by the end of the proximal convoluted tubule. So things move across the proximal convoluted tubule via a transcellular pathway, which is directly across the cell. They can also move via the paracellular pathway, and that's between the cells right here. And I realize there doesn't appear to be much of a gap, and there isn't much of a gap, but some things can squeak between the cells in the paracellular pathway. And one thing I do want to say is that while there's a great amount of reabsorption that happens in the proximal convoluted tubule, there is still some secretion that happens, and some things that are secreted in the proximal convoluted tubule. And to be clear, secretion is movement from the bloodstream, let's say from the peritubular capillary, into the lumen. These are things that didn't initially get filtered in the renal corpuscle from the glomerulus into the capsular space. So we can, or the nephrons can, the kidneys can, put things into the lumen, into the nephron that weren't initially filtered. And some of those things that occur or are secreted into the proximal convoluted tubule are waste products like urea, uric acid, creatinine. Some pharmaceuticals that people take will move into the nephron, and certainly the hydrogen ion. And we're going to see this along the nephron, specifically within the proximal convoluted tubule, distal convoluted tubule, and to a certain degree, the collecting duct. They will all secrete the hydrogen ion into the nephron, so the hydrogen ion can be eliminated from the body. The only way to directly get rid of the hydrogen ion is via the urinary system, by putting the hydrogen ion within the filtrate of a nephron so it can be eliminated in the urine. So I want to talk about the loop of Henle now. Keep in mind that the loop leaves from the proximal convoluted tubule. This is the descending portion of the loop of Henle, and this is the ascending portion of the loop of Henle. Descending portion is primarily made up of the thin segment. Ascending portion is made up primarily of the thick segment. Discussion of the loop of Henle is really the purview of a human physiology course. We're going to discuss it in brief here, but if you want greater detail, take a human physiology course, because a lot of it has to do with the process of osmosis, solute concentrations, which we've talked about, but it needs to be laid out in much greater detail than I am going to in the following images. Okay, so once again, the loop of Henle has a descending portion that is made up by the thin segment. It is thin because the lumen is lined by simple squamous cells that are mainly permeable just to water. It is not permeable to any solutes, any of the filtrate, only the water. Because the thin segment of the loop of Henle, or the descending portion of the loop of Henle, is not actively transporting or pumping things against their gradients, then there's no need for ATP, there's no need for simple cuboidal cells, and we want to set up, or the body wants to set up the opportunity for water to easily move across the tubule cells in the descending portion of the loop of Henle into the interstitial space and then back into the bloodstream. The ascending portion of the loop of Henle is primarily the thick segment, and there is certainly some active transport happening within the ascending portion of the loop of Henle, hence the need for the simple cuboidal cells with ATP and actively pumping and transporting entities back into the renal medulla, or the interstitial space. Once again, if you want more details on the loop of Henle and the production of the hyperosmotic renal medulla, please take a human physiology class. So let's take a quick look at the nephron once again. So one thing I showed you when we were looking at the proximal convoluted tubule were the tubule cells. So please keep in mind, when I'm referring to any part of this nephron, they all have tubule cells on each side of the lumen, on the top of the lumen or bottom of the lumen, depending on the orientation of the nephron, what part of the nephron you're talking about. The proximal convoluted tubule cells are just peppered with microvilli because that surface area allows for a lot of things to go back and forth. When we look at the distal convoluted tubule, it is lined by these simple cuboidal epithelial cells, but they have a smooth surface on the apical layer of those cells, or the apical surface of those cells. So there's certainly some reabsorption that happens in the distal convoluted tubule. I know in previous lectures I've talked about primarily reabsorption at the proximal convoluted tubule, primarily secretion at the distal convoluted tubule, and I will still hold to that, but things can be reabsorbed in the distal convoluted tubule. But once again, we can see, and we do see, a bunch of hydrogen ions secreted into the distal convoluted tubule and attempt to maintain pH homeostasis, as well as some other ions that can be secreted in the distal convoluted tubule. Now let's take a look at the collecting duct right here. The collecting duct, once again, is not part of any single nephron. It is shared by multiple nephrons, but this is the collecting duct, which is lined by simple cuboidal epithelial cells. And there are two types of cells that make up the collecting duct. We have intercalated cells and we have principal cells. The intercalated cells of the collecting duct are responsible for secreting either the hydrogen ion into the lumen of the collecting duct or bicarbonate. And we haven't really talked about bicarbonate in this class. And once again, bicarbonate is really more the purview of a human physiology class. It's really the other side of the hydrogen ion. But bicarbonate and the hydrogen ion can get secreted into the collecting duct via the intercalated cells. When I say via the intercalated cells, moving from the interstitial space or the extracellular fluid across the basal surface of the simple cuboidal intercalated cells, across the apical surface of these cells into the lumen, so we can excrete either the hydrogen ion or bicarbonate, and all of that has to do with pH homeostasis. The other type of cells lining the collecting duct, and certainly these are simple cuboidal epithelial cells, but a specific type are these principal cells. And the principal cells are responsible for reabsorbing sodium and water. Now, one thing I want to say is that water is always chasing solutes, given the opportunity. And when I say solutes, sodium is a solute, glucose is a solute, plasma proteins are a solute. Sodium is a huge solute, and sodium is something water loves to follow. So if the principal cells are reabsorbing sodium back into the bloodstream, water is going to chase that. So this is going to happen when the body needs that water. And when this happens, concentrated urine is produced. That is to say, the urine that ends up being produced is highly concentrated in solutes. Now, it's not going to be concentrated in sodium so much because a lot of that is being reabsorbed. And it's not so much there is a greater quantity of solutes in the urine. It's just that so much water has been reabsorbed that we produce, or the body produces, a darker urine because of the lower water content of that urine, because the water has been reabsorbed. So the principal cells are responsible for reabsorbing sodium and water. The collecting duct is influenced by two particular hormones. One is ADH, which is secreted by the posterior pituitary gland. ADH stands for antidiuretic hormone. A diuretic is something that causes the body or the kidneys to release water in the urine. An antidiuretic prevents the release of water or minimizes the release of water from the kidneys. So that antidiuretic is going to stimulate the reabsorption of water from the collecting duct back into the bloodstream. As a result, that's how concentrated urine is created. So the release of ADH from the posterior pituitary is going to stimulate the reabsorption of water through water channels known as aquaporins within the simple cuboidal cells of the collecting duct, specifically the principal cells of the collecting duct. Now, to be clear, conditions change throughout a day or throughout a week or month or a person's lifetime. There are times where we need to get rid of water. To get rid of water, we need to create a highly concentrated filtrate within the collecting duct to incentivize water to follow those solutes in the excretion down into the collecting duct, into the minor calyces, major calyces, and to be eliminated from the kidneys. And that is stimulated by a hormone released from the heart known as atrial natriuretic peptide. And the heart releases ANP, atrial natriuretic peptide, during times of high blood pressure. During times of high blood pressure, that can be alleviated by minimizing blood volume, and that can be caused by eliminating water from the blood plasma or the extracellular fluid. So, atrial natriuretic peptide causes the secretion of sodium ions into the collecting duct, into the filtrate of the collecting duct, causing water to chase it and be eliminated from the body, creating dilute urine. Okay, this image is beautiful. It is not my image. I've stolen this off the World Wide Web. And this is showing a structure within the nephrons and within a structure related to the renal corpuscle, the afferent and efferent arteriole, known as the juxtaglomerular apparatus. And this whole purpose of the juxtaglomerular apparatus is to regulate blood pressure. So what we have right here is the renal corpuscle with the glomerulus. This is a proximal convoluted tubule. This is actually the distal convoluted tubule. And I realize that seems crazy based on how I draw the nephron. But the way I draw the nephron, and the way a lot of books do it, is meant to simplify the different regions of the nephron. But I've showed you in a previous video how tortuous and convoluted a nephron can be, bending back upon itself. This right here is the afferent arteriole. This is the efferent arteriole. Truth be known, I'm not convinced that this is the afferent and this is the efferent, but I believe this is the afferent because it has more of these cells right here, which are known as juxtaglomerular cells, for short, JG cells. It doesn't really matter. My purpose here is to talk about the JG cells and another group of cells known as the macula densa. So lining this portion of the distal convoluted tubule, and I realize I may have said distal convoluted tubule twice. It's really the top part or the distal end of the loop of Henle as it's entering the distal convoluted tubule, this being the lumen. This portion of the tube is lined by specialized epithelial cells that create the macula densa. So this concentration of specialized epithelial cells creates a structure known as the macula densa. And these are chemoreceptors that are evaluating the filtrate moving through the nephron, specifically at the distal end of the loop of Henle at the beginning of the distal convoluted tubule. And mainly the composition or the concentration of sodium. This right here being the afferent arteriole is lined by specialized smooth muscle cells known as JG cells. These juxtaglomerular cells are mechanoreceptors. They are activated when there is a change in pressure in these blood vessels within the afferent or efferent arterioles. So the blood pressure is going to induce some vasomotion of these blood vessels by these specialized smooth muscle cells. But these smooth muscle cells are so special that they also secrete an enzyme known as renin. And renin is part of a mechanism known as the Renin-angiotensin system or mechanism that we're going to talk about in a second here. So what I want you to remember from the juxtaglomerular apparatus are the macula densa, which are chemoreceptors, and the JG cells, which are mechanoreceptors. There's a third group of cells known as the mesangial cells or mesangial cells, depending on the pronunciation. Truth be known, I'm not super clear on the function of these cells, so I'm not going to talk about them. Okay, lastly, let's talk about this renin-angiotensin mechanism, otherwise known as the renin-angiotensin system. And this is stimulated by low blood pressure detected by the juxtaglomerular apparatus. And the first thing that is going to happen is the release of renin from the JG cells that we just looked at. Renin is going to convert a plasma protein known as angiotensinogen into angiotensin 1. Angiotensinogen is an inactive plasma protein that only gets converted to angiotensin 1 in the presence of renin. Once again, renin is released from the JG cells during times of low blood pressure. When angiotensin 1 encounters another enzyme known as ACE, the angiotensin converting enzyme, it gets converted to angiotensin 2. Angiotensin 2 is a potent vasoconstrictor throughout the body. It causes vasoconstriction, which we want to happen when there is low blood pressure. Keep in mind, individuals with chronic hypertension may take ACE inhibitors, which will inhibit the production or conversion of angiotensinogen, or I should say angiotensin 1 to angiotensin 2, because we don't want vasoconstriction occurring in individuals with hypertension. But back to this system here, angiotensin 1 is going to cause blood vessels to vasoconstrict and attempt to increase that blood pressure. Angiotensin 2 is also going to stimulate the release of aldosterone from the adrenal cortex. And aldosterone stimulates the reabsorption of sodium. By reabsorbing sodium back into the bloodstream, it's going to incentivize water to be reabsorbed from the collecting ducts. Once again, water can only be reabsorbed if those aquaporins, the water channels, are present within the collecting ducts. And that is achieved by the release of antidiuretic hormone, which is also known as vasopressin. Antidiuretic hormone is also a potent vasoconstrictor. So, in review, the renin-angiotensin mechanism is initiated by the release of renin from the juxtaglomerular cells of the nephron, or specifically that are wrapped around the afferent and efferent arteriole, but are related to activity or the monitoring of the filtrate within the nephron. Renin is released during low blood pressure. Renin converts angiotensinogen to angiotensin 1. ACE, which is angiotensin converting enzyme, converts angiotensin 1 to angiotensin 2. Angiotensin 2 induces systemic vasoconstriction, causes the release of aldosterone from the adrenal cortex. Aldosterone stimulates sodium reabsorption from the kidneys. ADH opens up the aquaporins to allow water to chase that sodium back into the bloodstream. And ADH, once again known as vasopressin, also causes vasoconstriction.